welcome to the new episode of Drug Development Diaries. In this series of podcasts, we are discussing different chapters that are published in the book called Approved, The Life Cycle of Drug Development. This book talks about overall journey of drug development from research to patients. And there are different chapters covering different themes. And today on this particular episode, we are going to discuss cellular therapies. And let me welcome today's guests and let me welcome Naren sir. Hi everyone. Hello Naren sir. And may I ask you, sir, to please take this up and introduce our guests for today's episode and let the discussion going.
Yeah, thank you, Vihang. Thank you. Today we're going to speak to three exciting people who have written, contributed to this chapter. I know each one of them in different lights. I'll ask them to introduce themselves in detail, but let me tell you a little bit about them before we start. So Aalia Khan and Ankit Banik are students of IIT Mumbai, and they're doing their PhDs with Dr. Rahul Purwar at Immunoact. First of all, I find that just absolutely fantastic. I'm a big fan of this collaboration between academia and industry, and what better way to exemplify this work. So both Aalia and Anik, thank you so much for doing what you do and then coming here and talking to us. And Karan, I've known for the last couple of years, year or so, we've met in conferences and then we've chatted a little bit about various things. I'm going to ask him to introduce himself. But what I love about him is his like free spirit, so to speak, a little bit. You'll probably get some of that when we talk about it. So he sort of mixes music and science in a way that I just love. So maybe what I'll do is I'll ask each one of you to introduce yourself in a few words. Let's start with Aalia.
Sure, sir. So, hello, and thank you for having me on the podcast. That was a good introduction from Dr. Narendra sir. Thank you so much. I'm Aalia Khan, and I am currently a full-time PhD scholar at IIT Bombay in Dr. Rahul Purwar's lab. Before this, I completed my bachelor's and master's in biotechnology from Mumbai, which is where my passion for research truly began to take shape. Currently, I am working with CAR T cell therapies as a part of my doctoral research. And along the way, I have come to think of myself as an engineer of a different kind because I engineer immune cells rather than building bridges or machines. My ultimate aim is to make these living medicines, that is cellular therapies, widely accessible, so they can save lives across the world. That's all about me.
Niket?
Yeah. Hello, everyone. First of all, thank you so much. Thank you so much for inviting us to the podcast. Myself, Ankit. I am also a PhD scholar from Professor Rahul Purwar's lab. My background has been in a distinctly different field, that is microbiology. I'm from microbiology background, but that was like in the graduation period. Post that, my interest has shifted towards oncology as well as immunology. Right now, I'm working in the field of CAR, different from Aalia, CAR NK cell therapy, which is also a type of immunotherapy. And yes, of course, my aim is to promote equitable access of all these kind of therapies across the globe, and specifically in lower middle income countries such as India.
Thank you. And Karan?
Hello, ladies and gentlemen. Thank you very much for having me here. My name is Karan Gera. I was a molecular biologist by training. I used to work on lysosomal storage diseases during my research years. I used to work on mouse and dog models of those. In those early years, the idea was that we wanted to create more robust models so that they could be used for creating good preclinical data for genetic therapies. Over a meandering set of years, I worked in the fields of project management and business development after my research career. And all roads lead to home. And for me, the home was cell and gene therapies to very, very well begin with. And I joined Immunoact in 2023. And ever since then and before then, I've always been involved in bringing new therapies to the masses and trying to make sure that they're used in the right setting, and most importantly, improving and optimizing their outcomes, whilst at the same time keeping an eye on what must be done next.
Wonderful. Thank you. Thank you for the introductions. The textbook has a lot of information already, but just to get people excited about reading the chapter, maybe you can explain what cell therapy is about.
Okay, so cellular therapies in general language, basically, they are treatments where living cells themselves become the medicine. So we can say instead of giving a pill or an injection of drug, we basically take cells, often from the patient, and we either grow them out or engineer them outside the body to do a very specific job. For example, like finding and destroying cancer cell is one of the very known example. Now, the main difference from conventional drug is that these cells can easily adapt, they can interact with their surrounding, and they can even persist in the body for many more months and years. And that's how they act like a living defense system. So they don't just treat symptoms, but they can potentially reprogram the entire body so that they can fight the disease from within. And I think maybe that's why they are seen as the future of medicine.
Mm-hmm. So, curious, how did this come about? How does cell therapy come out? What is the sort of historical perspective of cell therapies, and where did they come from?
So maybe I would answer this question. So basically, if we go back to the history of cellular therapies, we have to go back to around maybe 1600 BC, where the Egyptian papyri they have the early records of trauma care or tissue repair, some things like that. Back in the about 17th century, there was like the discovery of circulations in the body. Early transfusion attempts were made. So like if we are talking specifically about cellular therapies, so we have to go back in the 1900s, where they have the idea, the mindset of people have progressed, where the idea of using living cells in the form of therapy was developed. So further in the 1950s, when we have also mentioned in the chapter, when we go to the 1950s, we have seen that the first successful bone marrow transplantation has taken place during that time. Gradually, there was graft versus leukemia effect was recognized post that period, where foreign donor immune cells were introduced into the patient body, and they could see the effect of those particular cells killing the tumor cells. When we are specifically speaking about CAR T cells, the concept of CAR T cells grew around late 1980s or 1990s by Dr. Zeli Eshar. So they have developed a particular re-engineered antibodies, where they have tried to show a binding of the antibody with a certain T cell receptors. And right now, if we see the evolution of our immunoengineered therapies or CAR T cell therapy, we can see that all these kind of therapies, the personalized medicine in the form of personalized medicines, they have come into success. The clinical success and the regulatory approvals of CAR Ts in blood cancers have been robust during the last few years. And so if we see that the effector cells, which are T cells, so people have also used different effector cells right now in the form of NK cells, macrophages, dendritic cells, and research is still going on. But yeah, if we look back into the history, it dates back to a very further period.
Wow. Now you give in literally 2 minutes, you've given us a 5,000-year history of cell therapy. But that's fantastic.
That's a gist of the thing.
Yeah. So then I guess the logical question is, what's going to happen in the future? So I know it's already cutting edge. So what is the cutting edge of cutting edge?
I think I should answer that. When it comes to genetically modified cellular therapies, you see all the gene modified cell therapies so far have always targeted B cells. They had easy targets. They had almost uniformly expressed targets. And that's the astute uniqueness of a gene therapy, or rather gene modified cell therapy. It's able to identify and then target a specific antigen. Now, we are lucky that in certain types of blood cancers, you have very obvious targets. And theoretically, you can live without them, so to speak, when you have the right clinical mitigation, such as intravenous immunoglobulin. But what about solid cancer? What about single gene mutations? So, luckily enough, we have a lot of innovations coming up here, which basically proceed on two different fronts. So, speaking about solid tumors, um, the understanding of the very complex tumor microenvironment, which is basically like a modified RNA, is much better than it once was. And therefore, not only novel biologics, but also genetically modified cellular therapies are able to identify these issues within the tumor microenvironment and de-risk them so far as to turn a cold tumor, inaccessibly penetrative tumor, into a hot tumor, which is a lot more penetrable.
Yeah.
At the end of the day, the immune system is only going to work so well as much as penetrability is concerned.
Yes.
So you need to enable that. So when CAR T cell therapies came across, they ideally included the properties for B cell and a T cell, the targeting potential of a B cell and the killing potential of a T cell. But what we're seeing now in genetically modified cellular therapies, especially for single gene mutation diseases like the mucopolysaccharidoses, like the lysosomal storage diseases, is that you're seeing that there are new designs of vectors that can incorporate, transduce themselves well into cells and replace or at least augment the genetic sequence of the otherwise incongruent cells and make sure that they produce the right protein or the right enzyme. And the case of oncology, what we're seeing is a development of combinatorial therapies where you're not only leveraging the immune system so as to defructify the tumor microenvironment, but also augment your response towards anti-tumoric activity. So better antigens, better targets, better construction of how you are targeting them, and better process development and manufacturing. That is a quick summation. And to the larger scale, what we're also realizing is they are not a singular output. You have to combine them with the right combination, with the right PDL1 inhibitor in case of say a solid cancer or a CTLA4 inhibitor, and most importantly, the timing is incredibly crucial. You do not want metastasis to have occurred to organs which are relatively impenetrable, like the bone or like the medulla of the kidneys. So timing is as important as the strategy of of cellular therapy, and that's what I would say for this much.
No, that's great. I think again, it's such a vast field. You captured the main elements of the innovative path in the forward in the unanswered questions, so to speak, and all of these are in the textbook. So thank you. So maybe I'll ask Gihang to ask a few questions.
Sure, sir. That's a wonderful discussion. We are all learning a lot about cellular therapies. Let us get back to the chapter that we have written here in the book, The Approved, the Life Cycle of Drug Development. Could you tell us a little bit about how this chapter is structured, what a reader should expect when they grab this book and go through this chapter on cellular therapies?
Sure, sir. So I think we structured the chapter to take readers on a journey. So we start with why cell therapies matter, then we dive into the history, the science, the regulations, and also we discuss in depth how they are actually made. I think Pratima has done a beautiful job in writing that part. Along the way, we have also looked at real world challenges, then the strategies to overcome them, and also what's coming next for the readers. So the overall idea was to keep it detailed enough for experts, but also clear enough that one doesn't need to be living in the lab, so that they enjoy the ride of working with cell therapies. And I also want to thank Ketki for beautiful graphical designs that she has put so much of efforts into designing so many figures in the. I think readers would enjoy seeing that part of the chapter as well.
Yeah.
Wonderful, wonderful. So overall, it looks like it was a very exciting experience for you to write this chapter. So could you tell a little bit more about it? How did this writing exercise shape your perception about this field? You all are researchers, or you are engaged in different activities involved to immunotherapy. So how has this writing exercise influenced your opinions and perceptions about this field?
For me, it was a great experience, to be very honest. It was more like putting the entire cell therapy world map, like proper map of cell therapy is kind of. So it was more like, spot the patterns, then you spot the gaps, then what are the exciting roads ahead. So that's how we have been through this entire writing experience. Apart from this, I think it has also sharpened our sense of how just game changing cell therapy can be. And it also reminded of one fact that explaining the vision is almost as important as the science itself.
Yes.
Great, great. Ankit and Karan, would you like to add something to this?
I
Yeah.
feel. Yeah, go ahead.
What I would like to add is, having been interfacing with the research community as well, or rather, to be more precise, with the preclinical research community, clinical community, uh, there was often a lack of handshake between the two. You see, in the United States and the European Union and all of these developed economies, it is interesting to see that these are physician scientists who develop these therapies. Fair enough. Good for them. It's great. However, in our context, our focus is patients. There are way too many, and they must be served. So, how do you create the right handshake between both parties? Writing this chapter was essentially trying to be able to communicate with both, whilst being able to say that, hey, these are what our learnings have been, and this is what our translation of those learnings has been. We need to find a way to make sure that patients can do better. Because by the time that they even have a hint of a moment of access to these therapies, might just be too late. So we wanted to make sure that it was lucid, that it gave them a good place to start. And even if they had absolutely no background in the modicum of genetically modified cellular therapies, they would at least have a benchmark, which is this chapter to begin. And Alia and Ankit did a phenomenal job of capturing the ethos of what has been done and what comes next.
Great, great. Ankit?
Yeah, so I actually have similar opinions to what Karan and Alia just said. I would like to add one more thing that in our daily this daily life when we are doing research and we are running and we are in a cat and mouse race, a little pause back and just a little introspecting into ourselves, how much we know, how much basics we have. We have advanced so much that we sometimes forget that what are the things which are lying in the past, the basics. So recalling all those things from the beginning till the end, including the manufacturing challenges, the regulatory things, the policy making, everything. So I think all those things have been, in one or the other place, has boosted our knowledge, as well as we have indulged in few things which we did not know earlier, and this particular collaboration has also allowed us to delve deep into into the cell therapy subjects which we did not have much idea previously about. So yeah.
Great, great, amazing, amazing. So just to follow up on the same part, Ali and Ankit, both of you are in your research career. So after writing this chapter, how has it changed your perception about your career in research? And similar to Karan, you are a professional already in the field. How do you think this chapter has impacted your perspective about this particular field and your career in the upcoming years? So maybe all three of you can take a small take on this.
So honestly, it felt like being thrown in the deep end of the cell therapy ocean, and there's so much out there to know about it. So it has definitely strengthened my professional outlook, I would say. Three big takeaways for me. First was that science is thrilling, but the regulations are the real plot twist, I would say. Second, tiniest technical details. It can decide the fate of a therapy. And the last one being the most important one, that every therapy has its own backstory of trial, errors, and persistence. So I think that's something that I have kind of learned while I was doing all the literature studies and writing, working on the chapter. Yeah.
Great. Ankit?
Yeah. So as a early researcher, I feel that getting all those details together, drafting the chapter, so many details, so many accounts to be considered. I feel somewhere our writing part, our knowledge, how to decode everything, how to bring everything together and accumulate it into a single place. That was that is a skill which one finds hard to achieve in one's lifetime. So I think that was somewhere that has added to our growth. And also, I feel maybe if I can suggest something to our juniors or someone, I would say that before doing any kind of experimental work, any kind of research work, I think first you should go through literature. You should see what is lacking in the literature, what is existing in the literature, and maybe through this through our chapter they can get so many insights into the cell therapy domain that they might be getting something new into their minds and proceed into the future directions.
Wonderful, wonderful. Karan, would you like to add something to this?
Yeah, I think so. Alia and Ankit and I often talk about our research strategies. By the way, you may all not know, but Alia is the innovator of India's first ever multiple myeloma targeting anti-B cell maturation antigen CAR T cell therapy. She
Wow,
is the
amazing.
innovator of that single chain variable fragment sequence and the larger perspective of how it would best be used. It is incredibly important to know that these are the people who bring about the innovations that we need. And if we do not have them from an academic perspective in our own country, they will not be approachable and feasible for most of the population that needs them. For me personally, as much as I am grateful that progress has been made in genetically modified cellular therapies for various types of tumors, I have a soft spot for the lysosomal storage diseases. And I was a part of many different patient-based societies. I used to spend a lot of time with patients as well, even here at Immunoact. Um, and I just wanted to make sure that the narrative, whilst still being a narrative for what has been accomplished in cellular therapies, what the current structure of manufacturing is, and what we can look forward to has been elucidated, was to bring a motivation to people to say that, all right, you know what? You worked on small molecules and you worked on novel biologics and genetically modified cellular therapies are the science of the future. The time is now. If we don't work on them now, if we don't bring patients the relief that they truly deserve, then what are we doing this for? Number one. And number two, um, unlike recombinant protein biological drugs, wherein most processes have pretty much become uniform at this particular point of time, right? In terms of scalability, cell line development, manufacturing. Genetically modified cellular therapies are still very heterogeneous. They're so different. Everybody's cellular therapy is different. Everybody's product is different. Everybody's platform is different. And yes, as Alia rightly pointed out, there are immense regulatory hurdles. But how long do we make patients wait? How long do we field queries from patients saying, I have a metastatic triple negative breast cancer, or I have a prostate adenocarcinoma, and I need a therapy now? China and the United States, the European Union, have made great strides on this. We are fortunate that we've had, people who've helped us through this entire journey, but we also know that there's a lot more to be done. And I just wanted to bring the perspective to people that there's a lot more to be done, and we need all of you.
Yeah.
We need all of you. And I guess that the chapter brings it about because Naren has done this his entire life. And everything that I knew about him was in terms of bringing accessibility to people. And everything that all the innovators who have made this chapter have done was to put that central message forth. And that's all I have to say about it.
No, thank you. So I think, first of all, as I said right in the beginning, you will see Karan's passion come out of this because they not only work in this, they actually live this therapy in their labs. So bring it to a close. Thank you so much, all three of you, for giving your perspective on this chapter. I think it'll be interesting for you viewers to put a face to the chapter as they read it. So thank you again for coming and talking to us about this. So I'll hand it over to Vihang to close the session.
Great. So it's been a wonderful discussion. Thank you, Alia, Ankit, and Karan, for your thoughts. Thank you so much for contributing to this chapter and coming here on board on this channel to share your thoughts. So to close down, to all our viewers, thank you so much for watching this episode. We will meet you again in the next episode on about a different chapter, about different step in the drug development process. Until then, look for this book. Definitely go and grab a copy, read this chapter, read other chapters as well. Tell us what you think about it in the comment section. If you want to know more about these concepts, leave a comment over there, ask questions to us, engage with this community of authors and researchers and scientists who have contributed to this chapter. Always stay curious, stay inspired, and I'll see you in the next episode. Thank you so much, and bye bye.